Assalamualaikum warahmatullahi wabarakatuh and good morning everyone. My name is Aina Shakira binti Idris. My student ID is 2024292978. So today I will be talking about the chapter 5 of immune system. Okay, so what is immune system? Immune system is a complex network of cells, organs, protein and tissues that protects the body from infection and diseases. So the first point of mine is the mechanism of innate immunity. So what is innate immunity? Innate immunity is a non-specific immunity. What is innate immunity definition is the body first line defense against anything harmful like bacteria, viruses or injury. It's called innate because we are born with it and works immediately and non-specifically. Meaning it's like a general and we already born since we are baby. So innate immunity provides the body immediate and non-specific response to pathogens, acting as the first barrier of defense before adaptive immune system is activated by Pradu 2024. For my second point is the component of innate immunity. What are the components of innate immunity? The first one is the physical barriers, the walls, which is preventing the pathogens from entering, such as skin, mucous membranes, and cilia, also stomach acid. The second one is cellular defense, which is we have phagocytes. We call it as eating cells. And the second one is the inflammation, antimicrobial proteins, and NK cells, natural killer cells. Okay. For my third point is the mechanism of innate immunity. What are the mechanism of innate immunity? The first one, we have the recognition of pathogens, where the immune cells have pattern recognition receptor that detects bacterial cell wall, viral RNA, and fungal cell wall components. The second one, we have the key cells that for the recognition of pathogens such as macrophages, neutrophils, dendritic cells, natural killer cells that produce cytokinase, which is a chemical signal to produce chemokines and trigger inflammation. When you get a fever, that's we call trigger inflammation. And the third one, we have the effector action where the phagocytosis, we have such as phagocytosis where macrophages and neutrophils engulf and digest the microbes. The second one, we have the killing infected or abnormal cells where NK cells kill virus infected cells and cancer cells. We don't want that in our body, right? So the third one, we have the complement activation where complement proteins punch holes in microbes, which is lyses, and tag microbes for phagocytes and attract immune cells. For my fourth point, we have the inflammation. When tissue is damaged or infected, blood vessels dilate that cause redness and heat. Vessels become leaky and it causes for swelling, and more immune cells enter that causing pain and pressure. Pathogens get trapped and destroyed to bring immune cells and destroy that contains infection to start healing. And the fifth one, we have the resolution and healing, which is the anti-inflammatory signal reduce the swelling and the tissue repair itself as the dead cells are cleared. That's all from me. Thank you.
Hi, my name is Muhammad Nazri. Okay, for this slide, I'm going to explain the first line of defense in our immune system. The first line of defense works before infection happens. Its main role is to prevent pathogens from entering our body. For example, our skin acts as a physical barrier that blocks bacteria and viruses from entering. Next, mucous membranes trap pathogens, especially in areas like the nose and the mouth. We also have cilia in the respiratory tract, which help to sweep out dust and microbes when we breathe. Teeth and saliva contain an enzyme called lysosome that can destroy bacteria. Besides that, stomach acid kills many harmful microorganisms that enter through food and sweat help stop the growth of bacteria on our skin. So overall, the first line of defense protect our body from infection at the very beginning. Next, this slide shows the second line of defense, also known as internal defenses. The second line of defense work after pathogens have entered the body. Its role is to destroy the pathogens. One example is phagocytes, such as neutrophils and macrophages, which engulf and digest pathogens. Another response is inflammation, which causes redness, swelling, and pain to help fight infection. Fever is also part of the second line of defense because high body temperature can slow down the growth of pathogens. We also have natural killer cells, which destroy infected or abnormal cells. In addition, complement proteins help to damage pathogens, and interference protect nearby cells from viral infection. So, the second line of defense helps our body fight infection internally once the pathogens manage to get inside.
Hi, my name is Muhammad Daniel Ashraf Barazali, and I will talk to you about immunological memory. So, what are immunological memory? So, it is a special feature in adaptive immunity and has the ability of the immune system to remember a pathogen to lead to a faster and stronger immune response upon re-exposure to the same pathogen. So, how does memory work? So, during the primary immune response, the antigens bind to the specific B cell or T cell, and after that, the clonal selection occur and activate the lymphocyte divided into effector memory, which can fight infection, and memory cell, which can life for future protection. So, what are the types of memory cell? Well, there are two, which is B cell and T cells. So, for B cell, it forms during B cell clonal selection and quickly produce large amount of antibiotic upon re-exposure to a pathogen. So how about T cell? So T cell is a long-lived antigen-specific T cell and can rapidly activate into cytotoxin or helper T cells. So let's talk about secondary immune response. So it is triggered when the same pathogen or antigen enters the body again, and the memory cell activated, producing a faster antibody, a higher antibody level, and a stronger T cell response. So, second immunity is stronger because memory cell already knows the antigen because it has been exposed once. So there's no delay for the first activation, and there are rapid cloning expansion of a memory cell or for the antibody. So it's more effective at target defense. So what is immunological memory important for our body? So for once, it is important because it provides long-term protection to ourselves because we will get exposed to many pathogens, to many antigens. So once we were exposed and re-exposed to the same pathogen of antigens, the body will already know and already prepared itself with antibiotics and memory cell. So it can also prevent repeated infections, and it is also the basic of vaccination because vaccines create memory cell without causing disease. Because vaccines are a weakened antigen that is put inside our body so that the memory cell can recognize it and be ready for the same type of stronger pathogen or antigen to come. Thank you.
Okay, so next we will move to role of lymphocytes and clonal selection process. But before that, let me introduce myself first. My name is Ahmad Ibrahim Bayhaki bin Abdullah and my student ID is 2024 28 3662. Okay, so next let's move to role of lymphocyte. So first of all, what is lymphocyte? So lymphocyte is a type of white blood cell that play a vital role in immune system, especially in defending the body against infections and foreign substance. So basically, lymphocyte is defending and protecting our body from various bacteria or virus, or the others. So the main role of lymphocyte is immune defense, which is lymphocyte help the body recognize and respond to pathogens such as bacteria, virus, and abnormal cells. And second is adaptive immunity. They provide targeted and long-lasting immunity unlike general immune cells. So next, let's move to types of lymphocyte and their function. So for the first type of lymphocyte is B cell, which is the function is produce antibody that binds to antigen. For the second is help neutralize pathogens and mark them for destruction. So B cell will mark the pathogen and then destroy it. So the second type of lymphocyte is T cell. So the function of T cell is coordinate the immune system by activating other immune cell. The second function is prevent excessive immune reaction and maintain immune balance. And lastly, natural killer or prefer to be called NK cells, which is kill infected or abnormal cells. Okay, next, let's move to clonal selection process. So, first of all, what is clonal selection process? Clonal selection process is a mechanism of the adaptive immune system that explains how lymphocytes respond specifically to an antigen. So, basically, clonal selection process showing us about how adaptive immune system encounter a threat to our body. So we have three process. So the first step is the body contains a large number of B and T cell or B and T lymphocytes, and each lymphocyte has a specific receptor on its surface. B and T cell has a different receptor on its surface. These receptors are different for each lymphocyte and are able to recognize only one specific antigen. For the second step, when an antigen enters the body, it will bind only to the lymphocyte that has a complementary receptor. This binding activates the selected lymphocyte. For the last step, after activation, the lymphocyte undergoes a clonal expansion where it divides repeatedly by mitosis to produce many identical cells called clones. So this clone cell then differentiate into two cell. So the first one is effector cell. Effector cell, which is B cell, become plasma cell that secrete specific antibodies, and also T cell become cytotoxic T cell that destroy infected or abnormal cell. So for the second one is memory cell, which remain in the body and provide a faster and stronger immune response if the same antigen enters again. So basically memory cell is just gonna remember the antigen and gonna respond due to the memory.
Okay, so we will start my part. So my name is Muhammad Nabil Hazwan bin Mahiruzani. Number ID is 2024 276 234. So in this slide, I will explain about process in cell-mediated and humoral immune response. So in this slide, I will show the cell-mediated immune response first. So first point is it is involved the lymphocytes, or we call it T cells, are the main players in this process. So it is effective against infected cells, abnormal cells, and intracellular pathogen such as virus. Next point is antigen presenting cells or APCs present antigens via MHC molecules. Helper T cells or CD4 plus activate other immune cells using cytokinase. This cytokinase T cells directly destroy infected or abnormal cells. Notice that this immune response does not involve antibodies. Okay, so next I will explain about humoral immune response. So if you guys saw in this slide, we have a lot of points in here and there's a picture in the right side. So if you can see, the first point is it is involves B lymphocytes or B cells. It is different from cell-mediated because cell-mediated mainly have T cells, not B cells. So for the humoral immune response, it is effective against extracellular pathogens such as bad bacteria and toxins. So the humoral immune response works. So the first thing is antigens bind to B cell receptors. Next, helper T cells activate the B cells. The activated B cells differentiate into plasma cells and memory B cells. So plasma cells is used to produce antibodies, while memory B cells can be for long-term immunity. So last, antibodies neutralize pathogens or mark them for destructions. So together with the cell-mediated response, humoral immunity provides comprehensive protection for the body. So if we don't have the cell-mediated and humoral immune response in our body might be attacked by pathogens or such as bacteria, virus, and everything that's dangerous for our body. So I think we can conclude that this topic is very informative for us to know, and I hope you guys be doing well and thank you for listening.